Okay, so welcome back to this next video in which we are discussing transduction of painful stimuli. Okay, right. So, we have now discussed the transduction of noxious heat stimuli and also the transduction of noxious cold stimuli that occurs within the axon terminals of nociceptive neurons. Okay? So we've discussed that what happens is you produce this depolarizing current in one way or another, which then causes this receptor potential, which is the depolarization of the electrical potential difference in the axon terminal, and then some of this depolarizing current will diffuse into the action potential origin portion of the nociceptor and will trigger this to fire an action potential, and it will then propagate up the nociceptor axon all the way up to the spinal cord. So, we want to continue now with this discussion by looking at the transduction of mechanical noxious stimuli. Okay, so let's turn our attention to the transduction of mechanical noxious stimuli, or of noxious mechanical stimuli. Now, of all of the transduction mechanisms that we are going to look at, the transduction of noxious mechanical stimuli is the least well understood. Okay, all over the place with regards to transduction of mechanical stimuli, whether it's noxious mechanical stimuli or not, it's not very well understood at all. However, what we think is that there is a channel on the end of nociceptive neurons, okay, like so. So nociceptive neurons which can detect noxious mechanical stimuli will have a channel on their ends, okay. So let's colour it in here. And this channel is called piezo2. Okay, so this is piezo2. There is a piezo1, but we don't think that it's involved in transducing noxious mechanical stimuli. Okay, we don't think it's present within nociceptive neurons. Okay, and basically when you deliver a mechanical stimuli, the idea is that this channel will open, and then the same sort of principle as always, it will allow a depolarizing current into the cell, so it will be permeable to cations most likely again. So for instance, sodium ions, calcium ions potentially, potassium ions maybe, and overall you'll get a net movement of positive charge into the cell, which will act as your depolarizing current. That will cause the receptor potential within the axon terminal, which will then spread to the action potential origin here, which will then cause the action potential origin to fire an action potential when this depolarizing current arrives there. Okay, right, and that's all I can pretty much tell you about the transduction of noxious mechanical stimuli. There's very little more than that known. Okay, right, so we'll turn our attention now to the transduction of noxious chemical stimuli, which is somewhat better understood then. Okay, so chemicals then. Okay, so we're going to start off by discussing ATP. Okay, so we've seen that when you get cell damage, okay, so let's say someone punches you, then lots of your cells are going to be damaged in that punch, okay, and some of them will lyse open and release ATP. So usually ATP is kept at a very tiny level within the extracellular fluid. Okay, so there's hardly any ATP within the extracellular fluid. However, cells have a lot of ATP within them. Okay, so this cell will have a lot of ATP within it. So when it lyses open, all of this ATP will be released out into the extracellular fluid, and that's a giveaway sign that there has been cell damage, basically. Okay, because usually there would be no ATP within the extracellular fluid. Okay, right. So basically, nociceptive neurons can detect ATP and will fire action potentials in response to it, and thereby they are detecting cell damage. Okay, right. So what channels then do nociceptive nerve endings have on them which can detect ATP? Well, these are what are known as P2x receptors. Okay, now P2x receptors are not quite common enough knowledge for it to be in the textbooks. So what I'm going to do is I'm going to give you a nice review that I have read and which I've used to source the information that I present here, so that you can go away and read that if you so wish. Okay, so the review that I read on P2x receptors is by a guy called Alan North, and it was published in 2002. Okay, right, but I think that what he says in here is still pretty much up to date, even though it was written 13 years ago now. Okay, so let's talk about the structure then of these P2x receptors. So, basically a P2x receptor has a trimeric structure, okay, and they are ligand-gated ion channels. Okay, so we've seen a lot of examples of ion channels in this video. However, not many of them have been ligand-gated ion channels. In fact, we haven't, I don't think, seen a single ligand-gated ion channel so far. All of the trip channels, trip V1, trip V2, trip V3, trip V4, trip A1, trip M8, all of those were activated by thermal stimuli, remember? Okay, yes, they were also activated by those molecules, capsaicin and menthol and things like that. But their main activation was given by thermal stimuli, okay? And you'll remember that trip V1 was not actually activated to open by capsaicin. What capsaicin did is it shifted the threshold for the activation of trip V1, okay? So it made it so that trip V1 would activate and open at normal physiological temperatures rather than at 42, 43 degrees Celsius, okay? So it wasn't really opening the ion channel, so we wouldn't have considered trip V1 a ligand-gated ion channel. Okay, right. So we're now going to see a ligand-gated ion channel, and these P2x receptors are all ligand-gated ion channels because they are ion channels which are usually closed but can be activated to open by the binding of a ligand such as ATP. So let's draw the ion channel here. So this cylinder represents the ion channel, and then down the middle you'll have the pore through which the ions can move. Okay, now P2x receptors are all trimers. They're made up of 3 separate subunits all joined together. So we've got one subunit here, a second subunit here, and a third subunit here. So basically these are not one protein, they are 3 proteins stuck together. Now, let's pull out one of these subunits of a P2x receptor here, which I'm highlighting in vivid purple, and let's have a look at its structure. Especially, let's have a look at its membrane-spanning topology. So I'm now looking at the subunit composition of these P2x receptors then. Okay, so here's the phospholipid bilayer again, and I should just stress that this side up here is the extracellular fluid side, and I want to leave as much space as possible for drawing it, which is why I've shoved that up there. And then this side is the cytoplasmic side. Okay, right, so let's now have a look at the membrane-spanning topology of the subunits of a P2x receptor. So basically the amino terminus of these proteins which are involved in the formation of a P2x receptor will be cytoplasmic. Okay, then you will have a membrane-spanning alpha helix which will take the polypeptide extracellularly. Then it's hypothesized that you have what are known as 5 cis loops within the extracellular domain. Okay, so let me show these. Like so. So these are basically loops in the polypeptide structure which are held together by cysteine interactions with other cysteines, okay, so by disulfide links. So I'll discuss in a moment what a cis loop is in more detail. So we have one cis loop here, one cis loop here, one cis loop here, one cis loop here, and one cis loop here. So this is a cis loop, 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 and this is a cis loop. So we've overall got 5 of them in this extracellular loop here. Okay, now let me discuss, as promised, what a cis loop is in a little bit more detail. So, This line will represent a polymer of amino acids, so it's representing amino acid after amino acid after amino acid after amino acid, etc. Now I'm suddenly going to draw one of the amino acids actually out, atom by atom, okay, and this is because this is one of these cysteine amino acids that's going to be involved in the formation of a disulfide bond that's going to hold the cis loop together, okay. So here is an amino acid actually drawn out. So, of course, it's going to be much bigger than the other amino acids because I'm actually drawing it out, but of course it would really be the same size as them. Okay, so here's the amino terminus bound to the carboxylic acid terminus of the amino acid in front of it. Here's the alpha carbon in the middle, and here's the carboxylic acid terminus bound to the amino terminus of the amino acid behind it. Now, that's the core amino acid structure. The R group, then, of a cysteine amino acid consists of a methylene group with then a thiol group coming off it. Now, a thiol group consists of a sulfur atom with a hydrogen atom coming off it. Okay? Now, I'm not going to show that hydrogen coming off the sulfur atom because I'm being fortuitous. I know that I'm going to have another cysteine on the parallel strand, which will be forming a disulfide link with this. And when they form disulfide links, they lose their hydrogen atoms from the thiol groups. So I'm not going to draw that hydrogen atom. Then temporarily I will go back to showing amino acids just as a line, basically. So this line now represents a polymer of amino acids. Okay, and then we'll come all the way around and we'll have another amino acid, which will again be a cysteine amino acid on this parallel strand here. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here is the R group, which consists of a methylene group with then a thiol group coming off it, okay, and here is the carboxylic acid group up here with then the rest of the polypeptide going off up there. Okay, and now what's going to happen is these two sulfur atoms within the R groups of these two cysteine residues are going to bind together, and that bond that you've got there between these two cysteines, that's known as a disulfide bond. Okay, so this is called a disulfide bond, or a disulfide bridge. Now, disulfide bonds are quite strong. Okay, so when you try to denature a protein, say you're doing a Western blot or something along those lines, and you try to denature a protein, what you do is you heat the protein up to around 85 degrees Celsius, and that causes most of the tertiary interactions that hold the protein in its folded state to fall apart. The problem is it doesn't cause these disulfide bonds to break apart, and they're the only ones which don't break apart, so they're the strongest. You then have to add some reducing agents such as beta-mercaptoethanol or dithiophreatol to break these disulfide bonds. Okay, you might ask, well, why can't you just take the temperature higher, rank it up higher? The problem is if you take it too much higher, you're going to start actually breaking the peptide bonds that are holding the amino acids together, and you don't want to do that. You just want to break the tertiary interactions. Okay, so it's quite a strong bond is the message from that little diversion. Okay, right, so why on earth do we think that these P2x receptor subunits have this complicated structure like so, with 5 cis loops within their extracellular domain? Well, the reason is that basically if you look at the extracellular loop for every single P2x receptor subunit, there is always, absolutely always, 10 cysteine atoms within the extracellular loop. Okay, so one here, one here, one here, one here, 4, 5, 6, 7, 8, 9, 10. And basically it's thought that these will all interact with each other to produce disulfide bonds, and therefore potentially it wraps itself up into this structure with 5 cis loops like so. But it's not yet been fully validated, okay? So maybe it looks like that, maybe it doesn't look like that at all, okay? But there is a good body of people who would think that it would look like that, okay? And would draw the subunits like that, hence. So, maybe it looks like that, maybe it's just an extracellular loop. Anyway, right, so basically these P2x receptors are trimers, okay? So they're made up of 3 of these separate proteins stuck together to make the whole ion channel. Now, the really simple thing would be if there was just a single P2x receptor subunit, and it was called the P2x receptor subunit, and all you had to do was make 3 of them, stick them together, and you've got a P2x receptor. Now, the reality is it's not that simple. Instead, there are 7 known genes which code for P2x receptor subunits. Okay, so there is the P2x receptor 1. Okay, there is the P2x receptor 2, and then it goes all the way down to the P2x receptor 7. Okay, so the naming system for P2x receptors is actually beautiful. It's very, very sensibly done. There is P2x receptor 1, there is P2x receptor 2, there is P2x receptor subunit 3, I should say receptor subunit rather than receptor, P2x receptor subunit 4, P2x receptor subunit 5, P2x receptor subunit 6, P2x receptor subunit 7. And they all have different sequences of amino acids. Okay, but they all share this same membrane-spanning topology here. Okay, so they all fold into something that looks like this. They all have the 10 conserved cysteines on their extracellular loop, which potentially form this 5-cis loop structure here. And they all aggregate together in trimers to form complete P2x receptors. So this gives us a huge plethora of P2x receptors that we could potentially build, because how do we aggregate them together to build the actual whole trimer? Well, one possibility is that you use homotrimers, okay, which it means that you join 3 of the same subunits together to make the full receptor. So for instance, you might pick P2x1, and then you might say, and now I shall stick together 3 P2x1s and therefore get a P2x1 homotrimer. Okay, and you can do that, okay? In nature, we have found homotrimers of all of the P2x receptor subunits bar 1. Okay, so the only one that has not been found to form homotrimers is P2x6. So, not. Okay, I'll just put there. So this one does not form homotrimers. All the others we found homotrimers of them within the human body. Okay? Or you could make heterotrimers. Okay, so homo meant same, so that meant that we were building trimers out of the same subunit. Heterotrimer means that we're using different subunits to make our trimer. It means that when we build this trimer, we're going to have multiple different types of subunit within the overall trimer. Okay, so generally the heterotrimers that you find only consist of 2 different types of P2x receptor subunit. Okay, so some examples are you can find heterotrimers of P2x1 and 5, which means that you can find receptors which contain both P2x1 receptor subunits and also P2x5 receptor subunits. Now, the stoichiometry, I don't think that's known yet. I don't think it's known how many P2x1s you take and how many P2x5s you take, because there are 2 possibilities. Either you could have 1 P2x1 and 2 P2x5s, or you could have 2 P2x1s and 1 P2x5. I don't know whether the stoichiometry has been figured out or whether both of those are possible. Okay, so then just think about how difficult it would be to actually work that out. Okay, then another example of an important heterotrimer, especially for us discussing nociception, is P2x2/3, which are heterotrimers made up of both P2x2 receptor subunits and P2x3 receptor subunits. Okay, so they have both P2x2s and P2x3s, and again, I don't know whether one of those stoichiometries is preferred over the other or anything like that. Okay, right. So, basically, you have P2x receptors within nociceptive endings. Okay, now the P2x receptors that you actually have are P2x3 homotrimers, okay, and also P2x2 2/3 heterotrimers. Potentially you also have P2x2 homotrimers as well. So the message is clear. You synthesize both P2x2 receptor subunits and P2x3 receptor subunits within nociceptive neurons, within nociceptors. Okay, you can then join them together to make homotrimers, okay, and mainly we think that P2x3 is probably more important than P2x2 homotrimers, and then you can also make heterotrimers where you've got mixtures of P2x2 receptor subunits with P2x3 subunits to make the entire trimer. Okay, right, so on our axon terminal now, so if this is our axon terminal here, we will have, this is the P2x receptor here. Okay, and basically when you have tissue damage, ATP will be released into the extracellular fluid, and these ATP molecules will go and bind to the P2x receptors. So you have 3 P2x receptor subunits, so you're going to get multiple ATP molecules binding to the entire P2x receptor, because they'll bind to each of the 3 subunits. Okay, what will then happen is the P2x receptor will open an ion channel. Okay, so that's why it's a ligand-gated ion channel. And for short, ligand-gated ion channels are often abbreviated to LGICs. L for ligand, G for gated, I for ion, and C for channel. Okay, so which ions then is the ligand-gated ion channel, which is the P2x receptor, permeable to? Well, basically, they're permeable to both sodium and potassium around equally. Okay, so the permeability to sodium is around equal to the permeability to potassium. And then they're quite permeable to calcium ions. The calcium permeability is very good. It's not as high as for sodium and potassium, but it is decent. Okay, so we know what this means because we've usually got an electrical potential difference across the cell membrane of around negative 65 millivolts. That's going to stop really much potassium coming out. So we're mainly therefore going to get sodium ions coming in and calcium ions coming in. So we're going to get a net movement of positive charge into the cell basically. And this is your depolarizing current then, which will cause the receptor potential, the depolarization of the electrical potential difference across the cell membrane of the axon terminal, and then the depolarizing current will diffuse into the action potential origin over here, trigger a depolarization of the electrical potential difference across the cell membrane there, which will then activate the voltage-gated sodium channels, and you're away with an action potential. Okay, and that's nociception complete there. Okay, so that's how ATP being released by cells which have lysed is going to trigger nociceptors to fire action potentials. The nociceptors have these P2x receptors on their surface, specifically P2x2 receptors, P2x3 homotrimers, and P2x2/3 heterotrimers, which will open and allow a depolarizing current into the axon terminal to produce the receptor potential, which then produces the action potential. Okay, right, so we'll call it there for this video, and in the next video what we'll turn our attention to is how an extracellular fluid acidosis, which means too high proton concentrations in the extracellular fluid, which is also a chemical noxious stimulus, is going to activate nociceptors.